guys are cool. My cheaper. Guys, this is so sad.
Like she spent like
Guys,
2 minutes just being like, well, the pH level in your brain goes down.
You know what got me messed She was like, we're not going to build it, it's a waste of resources.
All right, let's sit down and calm down and get ready to start. We have a lot of stuff to do today. You guys are responsible for the stuff tomorrow, so we need to do it. Can't wait, right? Yeah, I don't know what these are from, but we will put them all back.
You're responsible for everything.
You're responsible for everything ever.
Really? Not even Yes. A story dog.
Is
I was just like,
she was here?
Oh, sad.
I lost it. I was like, oh my god.
All right, guys. So where were we when we ended yesterday? Do you remember?
We were building models.
All right. So you heard something I said. That's good. The thing that mentioned candy. What?
We don't want M&M's, to be honest. Olive is saccharide.
All right. Good. So we've talked about. All right. I don't know if these animations are in order today. We'll see. All right. You're gonna fail this quiz?
Oh yeah.
No. All right. I thought the other day you said you're like I actually get this, right? No.
It was like 2 days ago.
Get it?
All right. So first day we talked about the first day on Monday we talked about how many saccharides. What kind of saccharide?
Monosaccharides.
Monosaccharides, right? And we classified monosaccharides, which is like one molecule, right? Based on how many what they had, how many carbons they had, right? So the prefix before an ose, right, refers to what? If we have a prefix
The
before
number of
ose,
carbons.
this refers to the number of carbons within one given molecule, within one monosaccharide. But if I use the ending saccharide, what is this prefix?
Number of molecules.
The number of monomers and the number of whole, molecules in it. So then this is the number of monomers or the number of monosaccharides in it, right?
See.
All right, so what's an example of something that has an ose ending? Hexose, right? So that's a whole category of them, which includes glucose and glucose, galactose.
Fructose, maltose.
Fructose is pentose.
No, fructose is a hexose.
Maltose.
All right, wait.
Ribose deoxyribose.
So now we have pentoses, which is ribose
And
and
deoxyribose.
deoxyribose. But you guys still recognize that all of these, each one is a monomer or a monosaccharide because they're sugars, right?
Yes.
Okay, and then what about ones that like have this, oh my gosh, these worms are over here. Have you guys like smelled when they come in and like change their
I've seen pictures of what
It's like
they
super
eat.
tangy smell.
Why is she doing worm lab?
Also, she's trying, I mean, it's biology, right? She's trying to keep, she gave them the task of keeping these guys alive, and they had to like research what they eat. And, anyway, we decided not to do that.
Yay. Thank God.
Alright, okay, so give me an example of the levels of saccharides that we're talking about. So these are all monosaccharides, and the main one that we're going to talk about really is glucose, right? But yesterday we went beyond this level. We talked about what?
Disaccharide.
So we had disaccharide, right? And what are the examples of that?
Lactose, maltose,
Lactose, maltose,
and fructose.
and sucrose. And what are they each made of? What's lactose made of? Rum.
Glucose and galactose.
Okay, what's maltose made of? Anna. Maltose. All of them have what in them? Glucose and glucose. Two glucoses, right? Glucose, and then sucrose is, Jasmine, glucose and fructose, right? So we're okay with this, right? We know disaccharides, right? What's the next level? So we did di, we did mono, right? Now we're going to do oligo. We did oligo yesterday. And what was the main purpose of oligosaccharides?
3 to 10.
George?
For the sugar, like to identify the cell.
To identify cells or recognition, right? Or for communication, cell signaling, right? All right, so we're just going to continue on that because we kind of got cut off. So this is where we're picking up. All right, so 3 to 10 monosaccharides, that's how many there are. This should all be a review. They are combinations of different ones. Remember, it's not just like a repeating unit, but it's like a different number of, different sequences of them, so it kind of carries a code, right? And then they are located on the surface of cell membranes. What do we say, on the inside or the outside? On the outside, and then the collective of all the oligosaccharides sort of extending out of the cell is called the glycocalyx, right? Which is essentially like the M&M coating, right? All right. And then we sort of zoomed in and we made this cartoony picture just so we could kind of appreciate that these oligosaccharides are made up of what each? What are these little shapes?
Those are monosaccharides.
Good. Monosaccharides, right? And notice how they're all kind of like different sequences, right? Okay. So then this picture we didn't have yesterday, right?
No.
We didn't talk about yesterday. Okay. So what did I do from this picture to this picture?
Enhanced down to the cell membrane.
I went even like deeper, so I zoomed in like way into here. So I'm at the level of the cell membrane, right? You see that? Here's the membrane. So this would be this. If we extended it, it would go all the way around here like that, right? So I'm all the way zoomed in on the cell membrane. And what do you guys notice here? You see anything kind of like interesting there?
Proteins.
So what I want you to notice is that thing right there and this thing right here. Right? So this thing, is it only an oligosaccharide by itself? No, it's also connected to what here? A protein, which is also another type of what? Not a carbohydrate.
Monosaccharide.
No, not a monosaccharide. Guys? Protein. What's the relationship between a protein and, let's say, like a carbohydrate? When we talked about the levels of carbohydrates, did proteins ever come up?
No, they're different.
No. It's a totally different type of organic molecule or biomolecule, right? Remember we said there are 4 different biomolecules. What are they? Carbohydrates, proteins, lipids, and nucleic acids, right? Okay. So notice what happened. So we attached an oligosaccharide, which is a type of carbohydrate, to a protein, which is a different type of biomolecule, right? And then we called it a glycoprotein, right? Okay, and what did we do over there? We took an oligosaccharide, again, this is the oligosaccharide, and we connected it to a lipid, and then therefore it's called a glycolipid, right? So the main takeaway message is that oligosaccharides don't just like float around on their own. They're usually anchored to a cell membrane by a protein or a lipid, right? And then they're called a glycolipid or a glycoprotein. Pretty simple, right? And then the sugar portion of it, right, of those like sort of mixed molecules, do you see that's an oligosaccharide, but that's also called, so what's the prefix that we use for the sugar part? Glyc. So the sugar portion, or the oligosaccharide, when it's kind of in a combined molecule, that's called the glycan. Right? So there are all these different words that are kind of getting thrown around, and I want you guys to make sure you're like really clear on what the difference is, right? So cocalyx is like just this whole collective layer of all the oligosaccharides extending out from the cell, right? The glycan it's basically like the sugar portion of a molecule, not the protein portion, but the sugar portion. And that portion usually tends to be a oligosaccharide, right? Of that level. Easy?
So
Yeah?
the glycoproteins, glyco part would be the glycan.
Yes. The glyco anything. So if I say,
or glycolipid,
yeah, if I said something like glyco teddy bear, right? The glyco portion of that would be called the glycan,
Okay.
right? Okay. All right. We're good? We're going to move on. All right. So does anyone know any examples offhand of oligosaccharide examples? No. Were you going to ask a question?
Yeah, so it surrounds the membrane.
So it's like basically all this, but going all around.
Yeah.
So this part is the glycocalyx here, this
Yeah.
little sugar part,
Yeah.
Yeah.
And it surrounds the.
the whole cell. It's like coming out of the membrane, like all parts, like radiating out of the membrane. Yeah. All right. So the first example I want to talk to you guys about is blood type, right? You guys have all heard about blood type. No? Yes.
Yes.
Okay. Do you guys know your blood types? I'm A positive too.
I think I'm AB.
So I could like donate blood to you if you need it. All right. So anyway, that whole ABO thing refers to actually an oligosaccharide that sticks out from your blood cells, right? Which is kind of cool. So people with O blood type have an oligosaccharide that looks like this, right? What kind of a mer is it? Is it a dimer? Is it a
It's a cool.
1, 2, 3?
It's a try.
Trimer, right? It's a tri. So it's an oligosaccharide, but it has 3 of them, right? So it's a trimer. Okay? So O has this. So basically, if you guys have a certain cell, do you want to attack your own cells? No. So anyone who, your body will recognize this as me. Okay, if I have this, I'm not going to attack it, right? So look, do you notice how this one is in here, and in here, and in here? Right?
That's why it's really.
So wait, yeah, okay, good. This is good. So anything that has this will recognize that as me. Okay? But then, so this is the oligosaccharide that comes out of O people's blood cells. This people who are A, right? They have this extra what? What is this thing? What are these little shapes? They have an extra monomer, or they have an extra monosaccharide extending over here, right? And then so A people, if they see this, or if they see this, they're like, okay, that's me. I don't need to attack that, right? But what if A people see this?
All right, that's cool.
They have it because they have that same sequence there. So they're like, all right, that's me too. That's fine, right? So they won't attack this. But what if O people, who are only used to having these 3 in their blood, suddenly see some blood that has these extra monomers right there attached to their blood cells? What are they going to do? They're going to attack it. They're going to be, that's foreign. That's not me. So that's why people who have O blood type can only accept blood from who? From whom? People who are also O blood type, right? What about B blood type, right? Look at this. So B has an extra monomer here also, but is it the same as A? No, it's a different one. So if you're B blood type, you have the same thing that O does. So will you attack O stuff? No, right? And you won't attack yourself, B stuff. But if you have something that has this yellow square in that position, will you like it? No, you're going to be like, that's not me, and then so you're going to attack it. So if you give B people A blood type, what's going to happen? They're going to attack itself, right?
Does
Question?
that mean that AB can accept any of them?
Yes. And so look at AB too. So AB has not only just like a mix of them, like not like halfway, but it has both. So it has ones that look like this. So there's some oligosaccharides that look like this, and there's some oligosaccharides that look like this, right? So that means that if they see the O, they're fine, right? If they see the A, they're fine. And if they see the B, they're fine, because they're used to seeing all these types of things, right? So the question is, so these are oligosaccharides, we already said that. So who's the universal donor? Who can just give blood to anyone and that person won't attack it? Lidu? Oh, right? So here we go. All right, and then who's the universal acceptor?
AB.
Well, AB, right? So you guys, is making sense kind of?
Yeah, but I don't think we're gonna get this involved, but like, what makes them negative or positive?
That's a rhesus factor. We're not getting into that.
Okay.
But like, look it up. It's interesting. But all right, so that's one example. I said to put 2 examples, right?
Yes.
So we'll go into one more example. Art.
So what's the main role of oligosaccharides? Is it like cell recognition?
Yes. Cell recognition, or you could say cell signaling. Yeah. All right. One more example, all right? And this is kind of interesting because it's like flu season right now, right? So on viruses, so here's the virus right here. What do you think these things extending out from the virus are? Do you think they're monosaccharides? Do you think they're polysaccharides? They're oligosaccharides, right? These are actually, these things that are coming out of here are actually, they're glycoproteins, right?
Oh yeah.
So that means it's really like, if we're going to go closer, here's a protein here,
Yeah.
and then here's a little sort of like chain sort of attached to it, right? All right. Have you guys ever heard of like the flu virus? How are they named usually? They usually have 2 letters associated with them. There's ABC actually of influenza strains. But have you ever heard of like H something N something?
H1N1.
Yeah, right? So this is, that's like, which one was that? Was that the swine flu or was that?
Swine flu.
That was swine flu? All right.
Bro, what? Yeah. I said next to him. Like,
All right. Okay. So basically, let me just give you the overview of this. On our cell, so here's the human cell, let's say, right? Here's our cell, right? Do you see these little extensions here? What are those, you think? Oligosaccharides. So it's really a glycoprotein also because it's anchored with the protein. But this thing is like an oligosaccharide or a glycan, right? And that's called sialic acid. Almost all of our cells have it, right? It's like this like sort of marker for our cells. So that's like this specific sequence on the outside of our cells that's just like hanging out there, right? Guess who can recognize that?
No. Virus.
Right, so what happens is there are 2 different glycoproteins coming out of these viruses. There's the H, hemagglutinin, right? That's where the H comes from, right? And then there's this other one called neuraminidase, which is the N part, right? So it's just 2, like if we were to zoom, so let's say these like, these pink ones are the hemagglutinin, right? And then those bluish ones are the neuraminidase, right? So there are 2 different types of oligosaccharides that are coming out of the flu virus. Which one, so we said the pink one is hemagglutinin, which one binds to the sialic acid and can recognize it? This pink one, right? So hemagglutinin, it like sticks to cells, right? It can recognize the sialic acid sequences and it goes and sticks to the cells, right? And do you guys know how viruses work? So basically what happens is were you going to say something?
Wait, is this like the same thing that we were like researching the other time, like a week or 2 ago?
Sort of.
Like, don't they like attach on and then they like take nutrients and then they like burst so they can reproduce?
Yeah, pretty much. So what they do is they need to first recognize the cells that they're going to attack. So who helps them? What helps them recognize those cells? Oligosaccharides, both on sialic acid and oligosaccharides on hemagglutinin, right? So it binds to the cell, it enters the cell, it starts to make copies of itself and like assemble those copies. And then where does it want to go? Does it just want to stay in that one cell? Where does it want to go then? It wants to spread to other cells so that it can infect all the other cells, right? So the role of neuraminidase, we said hemagglutinin binds it to the cell, right? But does it want to stay stuck to that cell forever? No. So neuraminidase actually helps these newly made ones detach from the cell membrane. So it kind of like cleaves it so that those new particles can then go infect other places, right? Have you guys heard of a type of medicine to kind of fight the flu? Tamiflu, right? You guys have heard of that? Tamiflu functions by blocking, it blocks neuraminidase, right? So even if the virus can still bind to the cell, the sialic acid, and then it can make copies of itself, but then if neuraminidase is like deactivated, what can't happen then? It can't leave the cell, so they're kind of all just like stuck there, right?
What happens like if you don't take enough or like you don't?
And if you don't take it early enough, it's too late because they've already gone and spread like exponentially. But that's why you have to take it like the day after. I think like the day of.
When the effects like wear off, can't they just leave again, or do they like?
Because by then you've made antibodies for it, so you're like, yeah.
What exactly is sialic acid?
It's just it's like a sequence that's just present on all cells,
Okay.
right? So most of them just have this hanging out there, like to recognize them.
Okay. I thought that was the oleo.
Sialic acid is a type of oligosaccharide
Oh, okay.
that's on the surface of cells to begin with,
Okay.
and that sort of makes them victim to the flu. Mike?
Are we gonna have to know like the hemagglutinin or
We should.
like for like the test?
You should know. You should know what these are. You should know that they're both what? All three of these actually. All three of these are what? Oligosaccharides, right? They're attached to a protein, but you should know them. You should know that sialic acid is the one on the cells, and the hemagglutinin binds it, and this one releases, right? And Tamiflu, like, disables this one, so that's why it helps you recover from the flu, right?
Wait, so what does the neuraminidase do?
So this is bound here, right? It wants to stay on that cell membrane because this thing likes those other oligosaccharides. But then when the virus particles want to go and leave and get away from the cell and go spread to other cells, neuraminidase will be like, all right, guys, let's go, and then we'll cut them off.
Cut off. All right.
But if they can't get cut off, then they'll just stay there forever. Okay? So Tamiflu again blocks neuraminidase. All right. All right. We're happy. Okay. So this is not happy. All right. Well.
Honestly.
All right. So now next level. We're gonna go to the next level. We're gonna go to polysaccharides. Right? Oh, there's way more. Do you guys remember what polysaccharides are?
They're like 10 or more.
More than 10, right? More than 10 what? Monomers, right? Okay. So let me just give you an overview of all of them, and then we're going to go into each one in depth. And then you guys are going to make your graphic organizers of it so you can process it a little better. All right. The first one is starch. You guys know what starch is. You've heard of starch before, right? Starch is a polysaccharide. And what's the purpose of it? Why do plants make starch? To store energy, right? Okay. So starch is the energy storage in plants, right? There are two types of starch, amylose and amylopectin. Just giving you an overview at first. We'll distinguish between them in a second, right? This is like the trajectory that we're going to go on today. All right. What's the energy storage in animals? And we talked about this already, so I hope you guys know this. Glycogen, right? When did we talk about this? When we were talking about what? Glucose.
Like explain how I should break things down.
Glucose.
And like the
Homeostasis, right? Remember when we talked about that? So like we don't want too much glucose in our body, and like if our cells can't use it for cellular respiration, what do? They store it up into glycogen, right? So basically glycogen is like the animal version of starch, right? Do we make starch? No, we make glycogen, right? Easy? Easy. Yeah. All right. Another example of a polysaccharide, which you might have heard before, is cellulose. Do you guys know what cellulose is? Or what the purpose of it is? So cellulose is present in plant cell walls. Can you guess what it might do? What
Makes it green.
makes it what?
Green.
Say green? No, those are chloroplasts. It's like this thick kind of material, and so it makes it
rigid.
It makes it rigid. It gives it structure. Good. So we have two functions so far that carbohydrates do: energy storage and structure, right? Have you guys heard of chitin?
Yes.
Yeah, sort of. All right, chitin is another polysaccharide, and this is responsible for structure also, but not in plants. In fungi, right, in their cell walls, they're not made of cellulose, they're made of chitin. And chitin is also present in like crustaceans, like in the shells of crustaceans, and in like the exoskeletons of like insects and beetles and stuff like that, right? So arthropods, do you guys know what arthropods are?
Aren't they bugs? Is it always
Bugs are arthropods. Arthro, think of like when you have arthritis, where does it hurt? Joints. So, and pods means like foot, right? So like jointed feet, like anything that has those like,
Oh yeah.
the like crabs and things like that, but also arthropods and stuff like that. Okay? Like insects. All right? So it's structure. So you see energy, structure, right? There's one more structure one, so it's called peptidoglycan. And it's for the structure of bacterial cell walls. So we have 3 different types of cell walls that have 3 different respective like polysaccharides that strengthen that cell wall. Said cell wall, right? Okay, and one thing so that's all there is. Easy, right?
Yeah.
But now we're going to go into the structure of each one of these. But you kind of get the overview of what their purpose is. How many main things are there? The main things that we're going to talk about really are just these 4, okay? But then, and these are just, you should know what those are for, like the function of them, okay? And remember we said oligosaccharides? What's the relationship of the different monomers within the oligosaccharide? Are they the same or different? They're usually like a different sequence, right? What about in polysaccharides? It's all glucose, right? So it's really, really boring. All it is just glucose, like repeated forever, okay? But the arrangement of them is slightly different. Okay, how far do we get? Am I answering all the questions? Number 12. What's the test for starch and what indicates? We'll talk about it in a second. All right. All right. So what is this? You guys recognize this?
The glucose.
All right, glucose, because glucose is the one that's making up all these polysaccharides, right? I want you guys to learn how to number them. So we're going to start from right to left, all right? And we're going to go in order, right, clockwise. So which one's going to be the first one? Oops. Okay. That, right? And then I want you to number them around. So 2, 3, 4, 5, 6. All right? That's really easy. All right, let's try another one. What is this?
Oh, no.
How many? We're going to start over here. Good. We're going to start over there. 1, 2, 3, 4, 5. So it's a pentose, right? Fructose is a... Is it a pentose? Fructose is a hexose. All right, so this is deoxyribose. And then I want you guys to number this one. I mean, this is as easy as it gets, right? 1, 2, 3, 4, 5. And that one is what?
Ribose.
Oh wait, no, there's
Ribose, right? So this is deoxy, meaning there's one less oxygen. That's just ribose, right? Are we okay? All right. This really isn't that hard, but you guys do have to sort of pay attention to the numbering, okay? So I want you to look at these. So I've numbered 2 of these. What's this?
That is it's glucose.
That's a glucose.
They're both glucose.
And this is glucose. Again, what are we talking about here? We're talking about polysaccharides. And what are polysaccharides made up of? Repeating units of glucose, right? All right, so this is what we did yesterday, but you didn't know what it was called because you didn't know how to number them yet. But now that you know how to number them, right, I want to make a bond between the first carbon of one molecule and the 4 carbon of another molecule. Right? So can you find the number 1 carbon on the left one and the number 4 carbon on the right one? Yes. All right, so we're going to circle them, right? Circle them, right? And again, what did you say, Arsh? What happens here?
Dehydration synthesis.
So dehydration synthesis happens here. So that is going to all come out. This O is going to go over here, right? And then we're going to have a glycosidic linkage or a glycosidic bond, right? But we're going to specify that by now calling it a 1,4 glycosidic bond. Why? Because it's between the 1 carbon and the 4 carbon. Don't forget that the oxygen has to be in the middle of them, but it's a 1,4 glycosidic bond, right? So the product of this is the bond, right, and water. But what process was this? Dehydration, synthesis, or condensation, right? Okay. Are we okay with this? Suppose I wanted to make another 1,4 bond with this and the next one over. You guys could do that, right?
Yeah.
So now I've made, once I have 3 of them in a row, what kind of saccharide is that? Then it becomes an oligosaccharide, right? And then once I add like 100 more of those, 1 to 4, 1 to 4, 1 to 4 bonds, then I will get a polysaccharide, right? And I've formed a polymer. So the process of adding more is called dehydration synthesis. It's also called condensation. It could also be called polymerization, right? Because I'm making a polymer, right? So we're okay? All right. So what shape, my question is, so here, I'm just going to give you a few in a row. And this picture is like, so the quality is so bad. But so I have now 3 in a row. And if I kept adding them 1 to 4, 1 to 4, 1 to 4, what shape do you think my whole molecule is going to be? Just straight, right? There's no kind of like zigzag or anything. It's just going straight across. Easy, right? Do you think that's it?
No.
No. So, if only. All right. So there's not only 1 to 4 glycosidic bonds, there are also things called 1 to 6 glycosidic linkages.
Wasn't that like the no,
Isn't that lactose?
it was something else. No, it wasn't.
We're still only talking about what? Glucose. We're still only talking about glucose. So even though maybe when we formed lactose, maybe there was a 1 to 6 bond, but I think most of them were still 1 to 4s. Fructose might have been like sucrose might have been 1 to 3, 4, 5. No, 1. No, I think it still is 1 to 5. 1 to 5 maybe. Anyway, all we're talking about is glucoses. So we want to make a bond between the 1 here and the 6 here. Can you find the 1 and 6?
Yes.
There, right? What's going to happen? So maybe this OH is going to go and steal that hydrogen right there. What's going to come out here?
H2O.
All right, and then what's going to form between the rest of that?
There's another H.
Glycosidic linkage.
So this O now lost an H, so it wants to do something, and then this C lost this OH here, so it wants to do something, so it's going to make a...
Glycosidic bond.
Exactly, so it looks like that.
But what about the other H2?
Yeah,
Doesn't that mean that
they're
another
both
H2?
connected to.
Well, if you just think of this, and like there's 1 H here, and then there's 1 H here.
Oh, okay.
So originally that looked like this. It was like CH, H, and then there was an OH, right? CH2OH is like this, right?
Oh, okay.
So then basically the CH2 just stays normal, and then this OH just goes and steals that H from the other, right? So this one forms a 1 to 6 glycosidic. We're okay with, you understand what's going on there? Okay, so suppose I have a chain of 1 to 4 glycosidic linkage. What shape was that again, Hannah?
A hexose?
What, if I have 1 to 4 glycosidic linkages, I have like lots of,
Straight line.
just a straight line, right? But then suppose I suddenly, along to that, I start adding 1 to 6 bonds. So here's my line, and then suddenly I start adding, so like here I have like my normal ones, right, 1 to 4. Suppose I add 1 here suddenly that has the 1 to 6 glycosidic linkage. What effect is that going to have on the molecule? So it's not only going in a straight line now, but now it's also going, so it's starting to branch, right? So the shape, when there are 1,4s plus 1,6s, it creates a branched shape in the molecule. Okay?
Does it still bond to the 1 carbon, or how does that work?
Huh?
How would it?
This 1 carbon over here bonds to the 6 carbon here,
Okay.
right? And then it kind of comes off, because then this 4 can also bond to another. So here, this one right here is a 1,4, right? This one's a 1,4. This is the one that's the 1,6, right? This one is also a 1,4. This is a 1,4. This is a 1,4. This is, So it's a combination of 1,4s that make it go this way, and then 1,6s that would make it start to branch. So there might be a 1,6 here, and then all these 1,4s here. And if we kept going down, we could have another 1/6 that would make it go up here. And then these might have a 1/6 coming off of there, right? So you see how like this one could, if this one had a 1/6 suddenly here, then it would make it go off again, right? So this just makes it branched, right? So it's no longer a single straight line. All right. Question, Georgia? Do you have a question?
Yeah, why does it matter which one's like a 1/6 and one's a 1/4? Like, does it make anything different or no? Like, if the one that made the 1/6 bond, why didn't it just like connect with the other ones on like the butt, the bottom one?
On the butt?
Like the one that had the 1/6 bond.
Yeah, this one right here.
Yeah.
Why didn't it just connect over here?
Yeah.
It could have, but it's just like the way it happens. I don't know. Like in animals, and you'll see which ones have the 1/6 bonds and which ones don't, right? But yeah, I don't know. It's kind of like a freak of nature that's just like the way it goes. All right. So starch, right? We said starch is what? What's the purpose of it again? Energy storage in plants, right? So and there are two types who are
Amylose and amylopectin.
pectin. All right. So amylose itself, really easy. It's a single strand. So what kind of bonds does it have?
1-4s.
Just 1-4s, straight up, and they curled it around a little here just to kind of make it pretty, I guess. But it's basically just like a whole string. Maybe the string gets a little twisted on itself, but it's still just one single line, right? Right. The other one called amylopectin, what do you think it has?
Branched.
Yes, slightly branched, only slightly. So do you see here's like the original like 1-4, 1-4, 1-4, and then here would be like maybe the 1-6, right? That causes it, and then these are also 1-4s that are just going 1-4, right? So amylopectin is slightly branched. So it has 1-6 glycosidic linkages, right? And 1-4, yeah. All of them have 1-4. Okay? And then, can I move on or not yet?
So you said slightly branched?
Slightly branched. Because it's not too crazy. It's just like, has a few 1-6s every so often. Right? And so, what was the test that we did the other day to test for monosaccharides?
Benedict solution.
You can't say it without saying the last part.
Benedict solution.
The Benedict solution test, right? And we were testing for monosaccharides, right?
Monosaccharides,
And
yeah.
what was the color of Benedict's originally? Blue. And what did it turn if there was a presence of? Brick red. Okay. Good. So there's also a test for starch, and unfortunately we're not going to do it, but you can know how it works. So it's called, I don't know why it's so small, sorry. It's called Lugol's, right? Lugol's is this solution, and it's basically a test for starch, right? And Lugol's is made of iodine. Do you guys know what color iodine is?
Is it like brownish?
It's like a brownish, rust kind of color.
Yeah.
Yellow in water, yeah. So, anyway, so when you suppose I mix something up with starch in it, right? If I mix something up that has starch, and then I add a few drops of Lugol's, does anyone know what the result would be? Would you say? Yeah. It turns like this purplish black, deep color. Blue-black, dark blue-black, dark purplish, right. So that's the answer number 12, right?
You have to do it in heat.
No. It happens like right away. Even when you guys know like this, do you think this has starch in it or not?
Probably not.
It does, because when you have the when you have the
Mike, stop.
when you have the if the iodine, if the Lugol's I can't stop. Just laugh. Okay. We're not laughing at you, we're laughing at I'm not laughing at you. Alright. Alright. So anyway, when I'm wiping up Lugol's solution off the table, what do you think happens to this? It turns black. So what does that mean is actually in here? There is starch in here.
How do you know there's starch in it though?
Like how do you like?
Because it's like from a plant, right? This is like paper. I mean, it was originally from a plant. Yes.
Did you ever go over what a glycolipid is?
Yes.
Same thing like
It's this fat that like holds it.
It's a glycolipid is like a molecule that's part lipid, part
like
sugar, oligosaccharide, right? And a glycoprotein is a molecule that's part protein, part oligosaccharide, or part sugar, right? Okay. All right, so we know the test for this, all right? What's the next one we're going to cover, you think? So we talked about starch, the two types, amylose, amylopectin. Amylose has 1,4 glycosidic linkages.
amylopectin is 1,4.
Amylopectin has 1,4 and 1,6. What do you think glycogen? So we're
A lot of 1,6. Yes.
all right. So glycogen basically is kind of similar to amylopectin, except it's like way more branched. So it has way more what? Way more 1,6 bonds, right?
But
So
it also has 1,4.
It also has 1,4s, of course. They all have 1,4s. So it's very branched, yes.
But it's still an amylopectin.
No, amylopectin is starch. Glycogen is in animals.
Okay.
So they're similar molecules, but it's not amylopectin. It's similar to. Like it's a storage molecule for sugar, but it's in animals and it's more branched. And then amylopectin is in plants and it's less branched. But they're both branched.
So glycogen only has 1 to 6, or it also
If it had only 1 to 6, what shape would it be?
Diagonal, like
it would
straight.
be diagonal.
It would
So-
go like up, like and it
Yeah.
would be, but it would still be sort of straight.
So it has so everything always has 1
1
to
to
4s, and then just some of them also have 1 to 6.
Yeah, and the more 1 to 6s they have, the more
Branch.
branched they'll be. Exactly.
There's not like a disease where someone can store glycogen.
There is.
Really?
There are. Yeah, you should look it up.
That just makes a lot of sense that would actually just.
All right, moving on, because I want you guys to get a chance to kind of do your models and your. Okay, so cellulose, we talked about that. Remember, was this for energy storage?
No,
For
structure
protection.
element.
For structure or protection, right? So cellulose, the structure is actually kind of interesting. So it's single strands. So that means what kind of linkages is it going to be? Single straight strands. So they're parallel, right? They're like in these lines. But what makes it so strong? Thank you. How did you know that? Oh, okay. So do you see how this is like a 1 to 4, 1 to 4, 1 to 4? One whole strand in it, right?
Great.
And then this one, again, what is it? 1 to 4, 1 to 4. It's like it's just a one single strand, right? But then what's happening between these? Hydrogen bonds. What's a hydrogen bond?
It's like when the hydrogen they like, you know.
So so there's going to be a hydrogen that's what?
Hydrogen is like slightly, like the polar.
Slightly positively charged, right? And then what's going to be attracted to that?
Oxygen. That would be
So there's going to be some oxygen here that's probably attached to something else that's kind of weak, right? So the oxygen's going to be kind of negatively charged, right? And then the oxygen's going to see this over here, and this, yeah, and so it's going to be like, oh, I want to kind of like make an association with that, right? So that's what's happening from here to here on all these. All those yellow lines are like the hydrogen bonds, right? And those are, like, again, we said one hydrogen bond can be broken pretty easily, right? One toddler, right? But then a ton of them, right? A ton of them would like knock you over, right?
Army of toddlers.
Right. An army of toddlers. Hmm?
One at a time.
What did I say?
I didn't realize what you were talking until you said army of toddlers.
Oh.
Don't worry, like take one of them at a time.
Take one and yeah, no, but trust me, like I have two of them and they're pretty sneaky guys.
Or weapons.
Alright, so okay, so here's do you guys see how this is like 1 to 4, right? 1 to 4?
Yes.
And then look at this. Here's an oxygen, right? What is that oxygen bonded to?
Hydrogen.
Hydrogen right here, right?
And the comp is at F1.
Oh no, sorry, wait. Yes. So this oxygen is bonded to a hydrogen, so it kind of wants, it's kind of negative, right? So then it goes and sees this hydrogen, which is also bonded to an oxygen. So this hydrogen is partially positively charged, and this oxygen is partially negatively charged because it's stealing all the electrons from the hydrogen. So this one's going like, oh, I want to. So, and that's what's happening over here. This oxygen is going to that hydrogen. And so, in any case, you don't need to know the exact molecular mechanism, but you need to know that they're hydrogen bonds, and that's what holds cellulose together.
I want you to convince me about this. Molecular love stories. Since they're already full, so they can't bond. So it's like Romeo and Juliet, but like on a molecular level.
All right. We're good? All right. What were the last two molecules? So now you guys, you have to know the structure of these. So you have to know that cellulose has the 1,4s, and it's like connected, the parallel strands are connected by hydrogen bonds. You have to know that amylose is straight. You have to know that amylopectin is slightly branched, and that glycogen is highly branched because it has a lot of 1,6 glycosidic linkages, right? Is anyone like totally lost? You have to know what each one is for. Some are for energy, some are for structure, right?
Yeah, I thought there was only one for structure.
Okay. So you have to know, that's, okay, good. We're talking about plants and animals, right? And then you have to know the two extra ones that are also for structure. And you don't have to know their actual, what they look like, but you have to know that they are for structure. So let me just take a look. All right? Shh. So chitin, we said fungi, cell walls, and arthropods, right? So this is what chitin looks like. This is like, it's kind of crazy, right? Because it has all these extra like weird amino groups in it. You don't need to know that, but what do you see that's maybe similar to another thing that's hydrogen bonds, right? So that could be like, that could be a hint for like the use of hydrogen bonds in biology, right? They actually provide support or structure, right? The other one that was for structure was peptidoglycan, right? Which is for structure in what kind of organisms? Bacterial cell walls, right? And then these, again, just look at the arrangement here itself, right? What's the arrangement that makes it kind of like strong and maybe durable? There are no hydrogen bonds here, but the parallel structure, if they're like kind of in this parallel form, that can also contribute to like a structure, right? If it's like a uniform structure, right? All right. So I'm just going to review, and you guys are going to tell me the answers here, right? So carbohydrate uses, right? Energy, right? Which one is used for energy? To like just to get energy from? Not to store energy, to get energy, to use it.
That's what I said.
Glucose. You break down glucose in cellular respiration to make what?
To make ATP.
ATP or energy.
38 exactly.
That's the first one. That's the main one, right? Okay. Next one. Shh. The next one was energy storage, right? What are starch in what? In plants and then okay, good. And what were the two types of starch?
Amylose and amylopectin.
Okay.
I know pectin. Amylopectin.
Okay. Structure. What are the three things that are structure? Cellulose in what? In plants, right?
Chitin in arthropods.
Chitin in
No, it's just plants.
fungi and arthropods, and then peptidoglycan in bacteria. All right, and what was the other thing, like the 3 to 10 one? What was the purpose of that? 3 to 10 monomers. That's oligosaccharides, so cell recognition, right? And that's the oligosaccharides on the surface of cells. And the examples of those were blood type and the flu virus, right? Or viruses in general too, yeah. We're good, right?
So good.
Alright, so I brought some poster board for you guys again.
Sick.
Do you guys know what a graphic organizer is? Graphic? Not just graphic in like the inappropriate way.
Splattered blood all over his face.
It's basically, I want you guys to make it. Do you guys know what a concept map is?
No. Yeah.
I can figure it out.
So what I want you to do, and I have instructions here, and you guys have till the end of the period to do it, and it's really for your benefit, and I want you to do it, I don't know, like in groups of 4 maybe? You think that's, the lab groups are good to do it? But so your task is, to take the 4 polysaccharides that we talked about today, right? What are the 4 that we talked about? The main ones. Polysaccharides. Starch, the two types of starch. So I want both types of starch, amylose and amylopectin, and I want glycogen, and then I want cellulose. So all four of those, I want you to make some sort of like visual depiction, and like the criteria are on here, but you need to kind of organize it on this board in a way that kind of helps you conceptualize it in a way. So you could put all the energy storage ones in one section and all the structure ones in the other one. You could put all the plant ones in one side and then the non-plant ones on the other side. You could put all the branched ones on one side and the non-branched ones on the other side. But you need to like do some sort of like division so that you're like sorting it out in your head. Okay? Yes.
For glucose and fructose, also energy ones?
Not exactly. No, glucose is really the main form that is used to break that. Yeah.
What's the difference between chitin and those?
You tell me. You didn't get it down. Can anyone answer that?
I got it.
George?
Peptidoglycan, it's like doesn't have any hydrogen bonds in it, and it's for bacteria cell wall.
Yes.
And then chitin one does have like some hydrogen bonds like holding them together, but they're both have strands that are like parallel to each other.
Okay. And then what's chitin for?
Chitin for like insects and fungi.
Yeah. So chitin is a structural support polysaccharide in fungi cell walls and in like insects and arthropods. Whereas peptidoglycan is another polysaccharide, but that's only present in bacterial cell walls. So they're divided, and no hydrogen bonds. But you guys don't really have to know about the hydrogen bonds in those. The main thing you have to know is like the hydrogen bonds, which are in what?
Hydrogen bonds.
Cellulose. Those are the main. Okay. All right. So come get a piece of paper, and then I want everyone to kind of like. So if you guys aren't at a lab table, I would like get at a lab table, right? So like you guys all work together usually or not?
Is it multiple choice, or like what is it tomorrow?
There's multiple choice and there's a fill-in section, so you might have to draw these bonds together.
So it's kind of like the last
Oh yeah. There's some fill-in parts. 1, 2, 3, 4. So go ahead and get a board.
Wait, like we have to like draw like glucose or
Look at the instructions. There's some instructions for how to
Yo!
approach it.
for your group. George, use the ABC.
For your group.
That's not very tactical of you, Art.
It's very tactical. Group do ABCs. Alcohol-based cleaning solutions. Your ABCs, you use ABCs.
Is it oil free?
That's a very bad.
Yeah.
Wow.
Right in your eyes. Or your ABCs. Alcohol-based cleaning solution. Excuse me?
I got alcohol-based poison on my baby.
I don't care.
by telling her to look at Omar because I thought you meant you were going to be doing nothing.
No.
I mean, Mike is reading at the table.
Also, if you guys deciding slouchy, realizing it.
Oh yeah, I saw you were like, oh man, you were like this, and I was like, I just saw a visual.
Oh, I know.
You were like Omar, and I'm like, yeah, Omar.
You started laughing and I
I thought someone was going to see me. So I would do this and then just like slowly go back. And you realize that it's taking like a good 15 minutes.
So wait, what do you want to do?
You guys want markers and stuff?
I'm back.
I was thinking
There should be like
Please put them back after you guys are done. If you're some rulers, I'll give you some markers.
Time to draw. Hexagon time.
Okay, here we go. Get 4. So you got like 17.
Here you go.
I know ambulance. We should put ambulance and ambulance because they are the starches.
Okay. Do you have?
She's gonna split this whole thing into 4 regions, and we can make it like a
So first, basically, we just have to
Alright,
Here are markers. You guys want? I don't have a meter stick. I have these little harder. These ones are a little more sturdy.
We got 3. Alright, set these up.
Yeah.
Yeah. Then trace it over.
Go by 7.
Might as well lay tap on the table. My leg.
Graphic organizer should have come on the side. Is he on the shiny side or the dull side?
There's not markers on this.
Yeah.
They get
markers. You guys want markers here? Purple markers.
Stop roasting. You can go a single conversation.
And then there's
I'm not
shiny here.
I don't care. I'm still.
You can just do each
Yeah.
Anyway, no, let's go with the exact half.
What are you guys doing, okay? Are you measuring how divided evenly?
Yeah, I think we're just doing
Yeah,
four,
that's
five, like,
It's fun doing.
Because our system is a bad word that
Like
starts
we're all
with
just
S
straight,
and
but then I
D.
work on like
What?
It's a bad word.
What did I do?
I was thinking something worse, but.
360. Alright, it's 360 meaning 180.
how about we do this and then spread like this? So you have to start. So we break starch here and then we break this and that. So it's like all the starch. Sarah's doing it. Sarah and I are telepathic.
Of course, Rose said. Alright, this is 260.
And then go down and then go down. What are you doing? What are you doing?
What am I doing now?
Yeah, we're doing it like this and then like this. So it's like all the starch and then like the maybe that's why you want like
On the way home, I got a boy.
Make sure you get involved in
You
this.
look like a sorcerer.
Process it all.
I can't believe it. Mans looks like a sorcerer.
Oh, we only have like 20 minutes, 25 minutes.
put your hat on.
So you wanna,
Alakazam.
You have to make it sound like it's like one of those.
Right, you know that this molecule is part of the spinning part of the
At least I know it's not bite.
Put your arms on the same level.
Wait, no, it's super bizarre.
Rodetta walking around and laughing with that in the morning was just like this. It's like almost on the floor.
Yeah, this is, when you did it, like, so you know that sugar is the only saccharide.
Yeah.
So, yes, I would make it, it's so long say 5.
It looks so.
Otherwise you might.
It's not complicated. I know, but it's too.
Oh. Why are your fingers so...
Jenna have one of those?
It's
Jenna.
Jenna.
Oh, that is Jenna.
Yeah.
It's like, really coincidental that like,
Wow. Okay, now we need to do this. I have a super. Are you gonna make this super exact? No.
Alright, go ahead.
guys, you don't have time to be too exact. I just want to see like the main things. So, is it kind of like, you know, kind of first reach?
Yes, just like you're going. It's
I'll take 80.
teacher.
I'll take
I need one person in the group that's like, make sure, you know, the leader, like the timekeeper, who's like, you guys, come on, this is the time we need to like get on it. Like,
15 minutes, you can do this.
Oh, yeah.
Somebody has to hold it. Good groups I know, like, right? So I need leadership, I need like, you know,
There's somebody that's
here, guys, no, we only have 25 minutes left. Come on, guys, so you need to do
25 minutes, come on.
four different things, so you have like 5 minutes each.
Oh, green's gonna be on the side, red is gonna be on the other side. Don't push me, bro.
Alright, good night.
I was scared that you think I'll get something.
It's right there.
You guys, when you make your glycogen solution, that light will always
Mike,
you know what?
I'm gonna fight you, bro.
Just like glycogen is an animal.
Sound like that?
I don't know what I
You want us to show the water? All weird people sound the same, okay? Because I can see you being like a white You know who You know who made cereal? Britain. So that's you know that. Yeah, they did. The US had a joint invasion.
That was probably like in 1880.
No, buddy, that was in 2003.
I don't really care.
Like Grandma was born in like the 1900s. No, wait,
No, but still, it happened.
I'm sorry, that is so not true.
Like, I don't know why you're. It's history, right?
Yeah, I don't know what you're trying to
You're
do.
trying to combat
I'm saying that I could be British because British made cereal in 2000.
The British in the 1980s had like they
came over in a boat like 1912.
The British invaded when they were trying to colonize the shit out of half the world. You know? Yeah, because like you can't win.
If you get nothing done, I will cry. Absolutely nothing. Exactly what's happening.
05.
I told Archie.
They want you to kind of like have some sort of code, yeah, for plants and animals. Yeah, that's very, very good. Alright, you guys have animals over here, plants over there?
Alright, am I doing this? Can I just do the line? Wait, what? What? I just don't that.
No, say plants dash. You like everything else. So no, you're fine.
I didn't realize that you were right there.
Look the paper.
Dash, animal dash. I can say you're plant.
Look the paper.
One strand. It's not that big deal. Wait, I got
And make sure you put everything.
You know what?
Don't do exact numbers. It's just free. It's just free. You have to do it.
I don't want like what I just asked not to do like this. Yes, but it's not straight. Yeah, it's not a straight line. It's gonna be otherwise that would be carbon. Wait, those are fine. Just write the picture and they just like we are. Sounds fine.
No, it's not good, but like I don't know if you do anything then.
Why?
Alright, so
Stop talking about it.
section and it says starch dash plants, right? So how are you guys
No self-hate. We can just restart. No time for self-hate.
No time to be down, come on. Then move on. It's fine.
That looks worse than mine.
No, it's fine. Okay. There we go. This will be annual pectin. This will be annual.
Just write it.
Annual pectin. Someone at the same time. So yeah. Now I'm gonna draw the model for like.
I'm gonna write the underlining. Wait, it's this.
Use one color.
That's an animal.
It doesn't matter. Just
No, do the same color. No, it's showing that they're the title.
That kind of hurted me though.
I don't care if it hurts you.
Envelope lighting is happening.
Literally, I remember one time in 7th grade there was a project and I had to always remember and like
Oh,
show
wrong.
back and you know nothing and I was like,
I've heard of them.
Wait. Envelope
and
lighting.
I ended up getting like 2. Anyway, I ended up
Why is it so fun?
absolutely nothing and I did all of
Are you related to Nina Rockenberg?
That's fine.
So instead of drawing the box,
No, Nina Rockenberg.
and then like all the actually got green.
no, what are we doing with red? We're gonna just like mistakes. We don't make mistakes.
No, you. Alright, just write dash.
Alright, so
Dash and then put it down. I'm gonna write dash and then plants.
it's what they're in. Okay. Like in the red? No.
Just do that with any. You can just.
I like blue. Okay, so we're gonna do dash what they're in.
So it's charts and they're held in by hydrogen.
Mike.
Like animals, right?
Yes. Let me also plants.
Unless you can use that
Maybe this name was
Animals or cellulose and plants, right?
Or like whatever.
It doesn't matter.
Yeah, look at mine.
Look at mine.
Did you do your plants? Wait, both of these are starch Why not?
So we have to write that in starch. Well, I mean, it's function is that it's a starch that
Can I
it's a starch.
Well, I know her name was Bland.
Cellulose is also in plants,
It's
right?
like a
Structure. Is this way? Is this right?
Wait, the function. Wait, just give in the function.
Plants and then structure.
I was really having a problem.
Did you say plants and then walls?
Yeah. No, the function is that's the function.
Okay, so underneath it we'll write function in red.
The structure of plants.
What's the function? What's the
She's
function
the
of
first
starch?
shot.
No. Just like you label
What's
it in
the function of a starch?
You know, they talk about it like every person's like, oh yeah, you're an accident.
Store energy in
Alright, they store energy.
My notebook is right there.
Envelope lighting.
There's a street structure.
Yes.
You know, the shape of my Tyler brand.
I have my notes over here. You can stop having acrylics.
Were there any 3 something bonds?
Then say, comma 4 is like.
Charles has your
Charles.
I mean white out. See what I'm saying?
Where is it? Yeah, that's the only thing that you're on. Wait, Charlie, I need a little one.
Right?
Little one. I don't cry, kind.
But then 3 is coming off. And this is where the number is like 1, 2, 3, 4, 5.
Mike, pick it up. Come on. Drop it.
We're gonna do the
Omar.
I found it on the ground.
Hard school basis. I knew what I was doing anyway.
You know what? Not
Wipe that out.
Oh, it's okay.
It's not what I said. It's tight.
Come on, are we on time? Can we get back to the target? Ours is finished in 10 minutes.
What's the difference between? What's the difference between?
It's the bond. But you guys didn't, so we're here now.
No arguing, guys. What's past is past.
Romo attacking.
Alright, are we on task? Are we on time?
You have a philosophy.
You have a philosophy.
I don't have a philosophy. Might just say if it makes good.
I can't say it's not water. Nobody knows why. You're not doing good right now. You're the one who's having panic attacks. Now I am. Okay, did you do it?
So, this is Billy. He gets all this.
You're all
I was just going off. And then, so like right now, he's like right here. Because he was
I got one of the first so shut up. Before I still hear in my head, right?
Yeah.
That one dropped was like right here.
There's always more. You have to show us a chain, right?
Another
Okay, wait. I'm doing blue and dot. We're doing blue or yellow. That's what I found. Just do like half. That's
That was so bad.
very simple to do that. But then I can't reach over you
I am
guys.
teasing. Do not.
I forget.
Like, you know what I'm saying? Sorry. Hater.
So,
We have like no time.
in 2011,
Come to this side.
So basically, in the political sciences, you gotta do the hand class.
When I ever brought up the point, so basically, so in the 2020 election, basically, so he, basically just said war.
And then you did this and so war.
do I think
I ran.
World War III, Iran, Iraq, bombing, like, that's literally all. Dude, I don't fucking post. What I'm saying, I wonder when I've ever brought up the point of the election.
Is this helping you understand a little better?
That's your.
Can I highlight this?
Yes.
6 months.
Oh, everyone
Let's
else
go
is a lot more.
multitasking.
You guys have like 3 minutes.
Why did you look
You guys
Yeah,
have
we're
10 minutes.
trying our best here.
It's very
I have politics over here.
Oh my god!
Oh, that sounds like
You did a very great job, remember? How many of you just... Yeah.
that was good, Olivia. Better than my upside down writing. Should I like label this where you're sitting?
Yeah.
And fix it.
Did you want 4 bonds? Did you do 1 bond? Yeah, that's a lot.
Outlining boxes with those.
It's only a 1 bond.
I got back to
But it has an O in
So
it.
you have 1 4 and a lot of 1
Try not to connect the lines with the O.
No.
Just like, okay, that's fine. It's just like, it's like, don't like that. Go like this. And then how you want that.
Okay, so it's 1, 4, and
So there's 1 4 bond. It's fine. How are you gonna make a 1 6?
Put 4 pencils down.
So here's a 6. You need to draw another. Thank you. Yeah.
You write
So
parallel for that
is
description. Can't we do another spell for the side? No, it's going on.
it has hydrogen bonds.
Parallel strands, P-A-R-A-L-L-E-L.
Right.
The hydrogen bonds is
That's right. That is right.
okay. So say these
These held together with hydrogen
imagine
bonds.
those 2 are meant.
Yeah, cellulose structure.
No, wait, watch what I'm doing, okay? So now here, so now I want to
They're not connected.
this one, you have a bond.
Olivia, parallel strands held together with hydrogen.
Here, hydrogen bond, you just have dashes in between.
See how I did that, right? That's why I thought you should make them smaller, so you have more room. Make another 1 4 bond over here, so it looks like it has like one branch and then another.
H-Y.
I want
T-R.
Like write the whole thing out like CH2.
No, you can write, no, but you don't even need to write the numbers as long as you're putting them on the right place. So you guys are like, oh, let's put them underneath. You can't, because if you put them underneath here, you could do, but you already had them on, you couldn't do a 1 6 here now, because they're together, right? You had a 1 over here, you could do a 1 to a 6 here. Right? And it's like, you don't really have enough room for anything, right?
Can I make it
That's why,
viral?
like, and there's a lot of people, the other kids too, are doing like, weird stuff coming off of their threes, right? But there's no bonds on there's no bonds on this is anyway, but it's going down. Yeah, so that's the problem. Okay. So here's your glycogen. Here's your glycogen. Here's your 1 4 1 4 1 4. You guys, it's not like that. Look, 1 to 4, 1 to 4, right? But now I want you to start adding like you don't have enough room here, your branches, and this one is going to look just like that. But I wanted it to show that you have more branches than that, right? So what I wanted to see is something like this. Coming up out of here. I wanted to see that like this was like an O, right?
Don't believe him.
Glycosidic link.
Like this, right? And then I wanted to see that this also has an O here, right?
Underline the field, Olivia, so you don't miss it.
That's all the words that are right here.
Sorry, I can't write.
You need better handwriting, and I
Yes, you can make another branch. And then here's like, so here's the main branch, right? Here's the main branch. 1 to 4, 1 to 4, 1 to 4. Here's a 1 to 6. Right? And here's 1 to 4, 1 to 4. And then I'm taking the other one here. Right? Show that it's like way more.
Why are your S's like that?
because
Right? That was like the idea.
no, when I write regularly, I'm sorry.
Okay, I was about to say. I was writing.
Draw one more down here.
Yeah.
I'm right?
Yes.
Do you understand it? Right. You understand what I'm doing? So I want, like, you can only add 1 and 1, right? And you can't add it to a 1 if the 1 is already
Yeah, that's good. The R is really good.
And you can't add it to a 2.
No, you have
add it to a 3. Right?
I think you guys need to take photos on your trees.
Oh yeah.
You can just make a ton of 1 to 4 ones coming like down.
Yeah, so I'm trying to trace this.
And what did you do?
Analyst is just.
Excuse me. I was telling you guys to do it.
I'm moving in 5, 4, 3, 2, 1.
And I gave you a pencil. I did so.
That's unconscious.
Thank you. Okay, ready?
I have the markers. We're like half done with the colors. We have this one too. I say in the same. Really want to
Miss Pittman, you choose not to. This, the amylose, is just 1 4 bones, right?
Just one for
Yeah.
Alright, what else? That's it. You want to transfer this? Alright, is there anything else? What is that, cellulose? No, I don't have anything else.
How about you use like a blue marker and you like go over the
You guys are going on four minutes here.
What is cellulose?
Cellulose,
It's cellulose is the bread.
So I should color red. Wait, before I mess this up. Red.
Alright. This is done. This is done. This is done. Yeah.
It's gonna be some president.
This is red. Honestly,
No, that's not how it looks like.
I'm asking myself that. Wait on this.
What else?
Mike.
Do that, that's fine. Like after you still have to show like for at least one sort of.
My handwriting also known as like my brother's handwriting, like
This one's already right. No, people lining it up.
What are you doing?
Okay, what are you?
You just draw the. You can draw
so you write it like so it's highly red.
Whoa,
You want to write it in Many 1 to 6.
Straight, yeah. Highly rich, yes. You go right upside down. Stop.
Which word is it, Sarah?
No one.
Okay, so what are we doing? Alright guys, how are we?
Doesn't matter, Addie. Addie, we're not going You're getting mad at me, okay? What are you doing? Make dotted line. What is that?
What do you need me to write? What do you write? Straight? Okay.
Draw